Hi, my name is Natalia Díaz-Arce, and the work I am presenting here today is a study about the genetic connectivity of Atlantic bluefin tuna. I work in the Marine Research Division of ASTI in the Basque Country in Spain, and this work has been carried out in collaboration with different researchers from other international institutions. Human fishing activities have an impact on marine biodiversity and ecosystems. The sustainability and conservation of those marine species that are directly or indirectly exploited by fisheries often depend on efficient fishery management plans. These efficient management plans involve a good knowledge about different biological aspects of the species, for example, for the definition of what are called the management units. Management units or fish stocks should correspond with natural populations, which share constant characteristics such as constant mortality rate or recruitment. Defining natural populations in the marine environment is often a difficult task, but analysis of the genetic profile of individuals can help to define such natural populations. For example, in the image we see a first example with the purple fish, which belongs to a single homogeneous population. The blue and the red fish belong to different isolated populations. These two populations, they might share a common ancestor, but since the moment in which both populations were isolated, they started to evolve, accumulating different genetic variations through generations until they became genetically differentiated. In some cases, these two stocks might mix in specific areas or different during specific periods of time, as we see in the dot circle below, and this should be considered for management of the species, particularly if fishing activity targets these mixed stocks. Now, let me introduce you to the case study of this work, the Atlantic bluefin tuna. This species is widely distributed along the North Atlantic Ocean and reaches incredible big sizes, in some occasions reaching more than 600 kilos. This emblematic species has suffered from heavy fishing activity to the point that it has been considered endangered and their stocks have suffered from depletion. Currently, the species is managed considering two different stocks, the western and the eastern stock, separated by the 45-degree west meridian line. These two stocks correspond to the known spawning grounds for the species in the Gulf of Mexico and in the Mediterranean Sea. Previous studies have found genetic differentiation between these two stocks. However, data recorded from electronic tagging data have found that if no individual has been recorded to visit both spawning grounds during their life, individuals that at least visit one spawning ground, they do cross regularly the 45-degree west meridian boundary line. So, to date, we don't know to what extent are populations of Atlantic bluefin tuna connected. On the other hand, previous studies based on genetic markers of this species detected that a small percentage of Atlantic bluefin tuna individuals saw mitochondrial DNA nearly equal to that of albacore tuna or Tunus alalunga. This fact suggests that hybridization between albacore and Atlantic bluefin tuna occurred, resulting in mitochondrial introgression in this species. Given the observed genetic differentiation despite regular movements across the boundary line, we studied the possibility of between stocks genetic differentiation being originated from this introgression process. On the top of that, some larvae and juvenile specimens of Atlantic bluefin tuna found in an area located near the western coast of North America in the cold slope sea constitute the evidence of previously unknown spawning activity in this area. However, the genetic origin of these individuals, whose finding was published only in 2016, is still unknown. Hence, with the aim of studying the genetic connectivity between populations, to evaluate the role of introgression from albacore tuna on stocks differentiation and determine the genetic origin of the samples from the slope sea, we sequence and obtain genetic information from more than 500 samples covering the 3 known to date spawning grounds and 3 different age classes: larvae, young of the year, and adult individuals. We found 2 different genetic clusters, which generally correspond to the Mediterranean Sea and the Gulf of Mexico, which are genetically differentiated. However, we also found some adult individuals captured in the Gulf of Mexico that saw a Mediterranean-like genetic profile. These are immigrants from the Mediterranean Sea. Also, the slope C, represented only by larvae and young of the year, saw an intermediate and heterogeneous genetic profile. Indeed, further testing for admixture between group pairs demonstrate that the slope C is admixed between Mediterranean and Gulf of Mexico populations. These values here. Inference of demographic history of the three spawning grounds based on genetic markers show that in one hand the slope C spawning ground was originated from the Gulf of Mexico relatively recently in the history of the species, and that in the other hand the three spawning grounds have been connected through genetic exchange represented by these arrows here. In this plot below, we can see estimations of contemporary migration, and the color bars, they represent the percentage composition of individuals' origin for each of the three spawning grounds. This way, the 68% of the Gulf of Mexico individuals could have a Gulf of Mexico origin, and the 31% of they would have a Mediterranean origin. Both analyses, they do agree in finding that the Mediterranean Sea acts like a donor population. Given that we observe genetic differentiation despite having evidences for high migration movements, we analyze signatures of introgression from albacore tuna among our samples as a possible source of genetic differentiation. We found that only Mediterranean and slope C samples, about 5% of them, showed an introgressed mitochondrial DNA from albacore tuna. We also found traces of nuclear introgression from albacore, again stronger in the Mediterranean Sea, weaker in the slope C, and no signal in the Gulf of Mexico. This means that this hybridization event probably occurred in the Mediterranean Sea, and then it propagated to the slope C through gene flow. However, the signal was not strong enough as to originate the genetic differentiation between the Atlantic and Mediterranean spawning grounds. As local adaptation driven by adaptive genetic markers can also maintain genetic differentiation in the presence of high gene flow, we seek for genetic candidate positions to be under natural selection. Genetic analysis, including only neutral markers, displayed the same genetic differentiation pattern, which means that differentiation is not or is not only driven by local adaptation. On the other hand, outlier markers group samples in three different clusters along the principal component 1 or x-axis. Along the y-axis instead, we can observe a structuring pattern similar to that observed when using all the SNPs. If we analyze separately those markers driving the differentiation along the x and the y-axis, we can observe two groups of markers that are under linkage disequilibrium. This means that those markers are located within genomic blocks that are inherited together. Indeed, the group 1 markers, those that differentiate the three cluster pattern along the x-axis, these ones here, they show characteristics found in chromosomal inversions. Chromosomal inversions are mutations in which a chromosomal fragment is inverted, as we see here. Those individuals on the left of the plot could carry two copies for the inverted version. These ones could carry one copy of the inverted version and the regular version, and these ones here, they would carry two copies of the regular version. If we analyze proportion of individuals carrying this inverted haplotype, we observe that it is more common in the Mediterranean Sea and than in the Atlantic populations here. Interestingly, when we analyzed both groups of genetic markers under selection, including albacore tuna, we observed that samples are polarized, placing albacore tuna next to the Mediterranean samples and next to those carrying the inverted allele. This means that those variants that are under selection are inherited from albacore tuna through an introgression process. This is interesting because both introgression and chromosomal inversions are known sources of adaptation in different species, and in this case, even if local adaptation does not drive genetic differentiation between Atlantic and Mediterranean stocks, this might constitute an important element in the species' biodiversity. To conclude, our results show that the Slope Sea is a recently originated spawning ground which receives genetic inflow from the other two spawning grounds. We also saw that the Atlantic spawning grounds are connected with the Mediterranean population through unidirectional westward gene flow. Finally, we detected introgression from albacore tuna, which segregate in different proportions among the three spawning grounds. While this introgression is not the main force generating genetic differentiation between Atlantic and Mediterranean spawning grounds, it is a potential source of adaptive features and helps understanding the genetic connectivity of Atlantic bluefin tuna. This work leaves the door open for some hypotheses. So after we discarded local adaptation or genetic introgressions as the main isolating factors generating genetic differentiation, another plausible reason for what we observe is that the effect of this unidirectional gene flow has been increased only since recently. This increase could be caused by changes in population sizes, and indeed, some decades ago, both the western and the eastern stocks collapsed. Indeed, during the last years, the Mediterranean population has recovered much better than the western stock, which still remains actually not fully recovered. Alternatively, this increased migration could be due to oceanographic changes that might derive into more attracting conditions in the western Atlantic. As a consequence, Atlantic bluefin populations could be under the risk of genetic erosion, which might have a relevant impact on the conservation of the species. So that was all, and thank you very much for your attention.